Method 3, minimum evolution. Related to the others, but instead of just arbitrarily joining things together because they are the closest, regardless of what that may do to your tree, we are instead going to look for what is the minimum number of molecular changes that we can make, so either nucleotide or protein changes, that will connect up all the branches in the tree. I think this method makes a lot of intuitive sense to a person, because when you're doing this method, you're actually making a hypothesis about what the genome or proteome or protein looked like at every step in evolution. And if this evolutionary hypothesis is right, there was actually a genome at every step. Have you reconstructed it correctly? Probably not exactly, but are you close? Maybe, yeah. So yeah, that's where we're going next. Minimum evolution tree. So, all right, let's get into it. So, main assumption is that you want to minimize the branch lengths, and so it's going to be a question: how do we do that? So, lengths are going to be computed from the distance between two sequences. So, kind of like that. In that way, it's going to be similar to the parsimony method, which we're going to cover in a minute, which is shortest branch length only, but not implying anything about how we got there, not implying anything about the evolutionary process. Yeah. So, minimum evolution is available in the Clustal W package or in Phylip, yeah, among other things. You can do a neighbor joining tree, you can do a minimum evolution tree, and you can compare the results, see how they're similar or different. With only two sequences, you will not see much of a difference, only in the links of the branches, maybe, but probably not even. But when you have more and more sequences, then you can start to see some groups will kind of detach from one tree and move around to another part of another tree. They'll usually be grouped because they're close to each other, but where they join on to the rest of them will sometimes change, and it's kind of neat to see that. This is where the uncertainty in these trees comes in. All right. So, why might we want to do a minimum evolution tree? Well, they're easy. You can do them online, yeah, which helps very quickly. Yeah, they work well for sequences that have high similarity scores. So you're not considering sequences as sequences per se, you're just looking at the distance between them. So you're potentially losing a little bit of information because we're doing this kind of mathematically instead of doing it with, sort of real nucleotides, if I remember correctly. And we're treating all sites equally. So remember the zinc finger example from earlier, where we said, oh, these four cysteines are always there, so they're probably important. They might be working together. They might be a zinc finger. In this case, we're going to treat each of those C's exactly with the same weight as the things next to it, even though some things often change and others don't. That's why the similarity matrix type approach with the hidden Markov models, I think, is superior, at least when you're dealing with quite different sequences. So for things that are pretty close, this is a great method and has a lot to tell us. Yeah. It doesn't always work well for distantly related sequences or divergent sequences. It's basically really dependent on the quality of the alignment that you're able to get. And for most people, what they will do is they will take an alignment, and if the sequences in the alignment are more than, let's say, 30% identical, that's okay. But if they are less than 30% identical, then you have to go into that alignment and look at it and make sure it makes sense, and maybe manually adjust it to take out some gaps, or just close things up, say, oh, we know this is the active site, so that should definitely be aligned, stuff like that. Yeah. So here we go. We're doing parsimony and maximum likelihood. Now, these are both character-based, so we're going to treat our nucleotides or amino acids as if they are characters, as if they are nose length and, ear shape or whatever, and you're just giving it a number. It's a type 3 nose, it's a type 5 ear, yeah. What's the distance between 3 and 5? No idea. They're just different things, so they're not closer or farther from each other. That's what they mean by it is not really using the sequence information. So it doesn't know about transitions and transversions, but if you're in a situation where those are going to be similarly distributed throughout your data set, then it's okay, yeah. It's just that you're making that assumption when you're using like a maximum likelihood kind of method. Okay, maximum parsimony, let's go. Parsimony, remember, is cheapness, and so we are just looking for, we're making a bunch of trees, and we're looking for which of those possible trees is going to have the shortest branch lengths total. It's a really old method, 1966, second oldest one we've covered, and it can generate phylogenetic trees based on morphological characters like eye color, bone length, number of teeth. Or it can do it on base. So at amino acid 1, it's an A, or versus at amino acid 1, it's a C. Yeah, something like that. Okay, and so you can infer relationships between things using this. So here's a nice little tree of some globins. What you see on here are distances. So this is the genetic distance between these things. And so genetic distance should be proportionate to branch length, and you want to make the minimum branch length for the entire tree. And so in this case, arrow 1 indicates where we think the root would actually be, so it would be equally related to all the globins, including plant globins and insect globins. And arrow 2 would be the last common ancestor of both insect and vertebrate ones, with the last common ancestor between plant and vertebrate ones would be over here. So this, 1 was the last common ancestor of the vertebrate globins. Yeah. So in maximum parsimony, we're going to say fewer changes are preferable to more changes. So shorter branch lengths are preferable. Did evolution actually take that path? Probably not. But we're trying to find out what is the simplest possible solution, like the quickest way it could have got here. And if you have a case where evolution maybe for a while needed a particular amino acid, then it needed another one that was now favored, then this other one was favored, and it goes back and forth. Yeah, you can jump back and forth between states. So evolution doesn't necessarily work this way, but if you really zoom out, and we're saying we don't care about individual steps, we just care about long-term changes, then I think it makes a certain amount of sense. So we're going to assume that these all these sequences exist in some kind of nested hierarchy of relatedness. That's like in the previous example where we had the parentheses saying these guys are in a little nest together. They're the closest. And then if we put the next bigger nest out there, it would be these two plus this one. And so now they're in a slightly bigger nest together like that, so that they're all inside, the way the other tree looked. And then we're going to say that the most parsimonious group is going to best describe the relations between those genes that were derived from common ancestors. So we have to use this with homologs. We cannot use this with things that are not homologous. And so you need some kind of outside support, some kind of number from some kind of tool that says that, yeah, within a reasonable range of confidence, I believe these are homologs. And again, that's where the interpretation comes in. And that's why I hammer that we have to do interpretation with every one of these assignments. Because, yeah, garbage in, garbage out. If you put in bad data, You will get a bad tree, you will make bad conclusions, and if people make decisions based on those conclusions, those decisions will also be bad. You see stuff like this all the time with the hunt for the origin of SARS-CoV-2, the COVID-19 virus. There are a lot of different groups that are coming up with a lot of really freaky, weird hypotheses based on very thin data down at the bottom of the tree. They're like, oh, these branches join, therefore there were 2 infections at the same time in different parts of Wuhan, or there was 1 infection that started out the whole thing, or yeah. It's really hard to make those conclusions when you have a lot of things that are very close. You know how those are related, but the next nearest sequence is very far away, which is currently the case with SARS-CoV-2. So it's a question of where does the root join onto your tree, and rooting, there's an art to rooting a tree, and it is not simple, and you've got to know how much confidence you have in your root. And I don't know of any tool that will just spit out and tell you this is a good root, that's a bad root. At the moment, this seems to be something that needs to be done by eye, by feel, and people have different feelings. That sort of thing makes me kind of nervous. So yeah, that's one of the problems with these sorts of tree building methods. All right, so we're going to pause there, and then we'll come back and get into it a little more with maximum parsimony.